Well, good morning and good afternoon for both of you in the East Coast and West Coast, and also everywhere else in the world. My name is Max. I'm the co-chair of the EMGS Bioinformatics Challenge 2022. I welcome you all for joining today's event. And with me, I have Jeff, who is also a co-chair of today's event. So today we have three exciting teams and speakers who are going to be talking to us about three tools for bioinformatics application in toxicology, and we are really excited to hear about their presentation. Now, just to go over the format of today's presentation. So each of the speaker would have 15 minutes to present their work. It will be followed by a question and answer section for 8 minutes. The question and answer section will be held after each presentation. Also, both the audience and judges can ask questions. And basically, what we are going to do is we would let the judges to ask their question first, and for the audience, if you guys want to ask a question, please to put your question down in the chat box. So both myself and the judges will go over the question in the chat, and then the presentation will be evaluated by 4 criteria, which include the model performance and the identification for a novel genotoxic modes of action, the effectiveness of data visualization, originality and innovation, as well as the speaker performance in response to questions. Now, finally, I want to thank the participation of our judging panel. Without them, we won't be able to host this event. Really appreciate their help on this, and absolutely the participation of all the presenting team. Really excited to hear your presentation. So with that, I would like to introduce our first presenting team, Dr. Rupen Bhattacharya from the University of Michigan. So Rupen, take it away, and thank you so much.
Thanks for the introduction, and I will just start sharing my screen and my presentation on. Okay, I hope all of you can see my slides in the full screen mode.
Yes, perfectly, thanks.
Yeah, and I hope all of you can also listen to me clearly. So if there's an issue in between, just let me know, and we can take care of that. So hello, everyone. I am Rupam. I am a 5th-year PhD candidate at the Department of Biostatistics, University of Michigan. And today, what I'm going to present before you is effectively the 3rd chapter of my PhD dissertation work. I have my dissertation proposal coming up real soon, like the next Tuesday. So it was good to have this presentation because I have to make a few less slides. I work with Professor Veera Baladandevatapani and Dr. Nicholas Henderson, who are my co-advisors. And this work is co-authored with them. The work has a meta-archive version that you can see on top here. And it's already published in the Proceedings of the Pacific Symposium on Biocomputing 2023. If any of you are going to PSB, I'll be presenting my work there on 5th January next year. So with that out of the way, I'd like to begin with what the focus of today's talk is. So my thesis broadly focuses on precision medicine in cancer and how we can use Bayesian statistics, Bayesian machine learning methods in those contexts. And obviously, as you all definitely know, there's a huge stream of data that you can get, not just for cancer, but for many other diseases. There are focused databases nowadays where you can get multi-omics, multi-clinical, and other dimensions of data across multiple variants, biomarkers, drugs, and so on. And my talk today is going to obviously focus on diseases, in particular cancer. We are going to look at the genomics of such disease and how they affect the particular proteogenomic and other kind of biomarkers that can affect the progression of the disease and survival of patients and so on, and also how those markers can make a person or a model system react to drugs or therapies in certain ways. So we're sort of trying to combine multiple dimensions into a single modeling scheme here. That's why this is integrative. So the motivation of this work comes from how cancer databases that are available around us work. So there are multiple patient and model system databases. TCGA, the Cancer Genome Atlas, is one of the largest patient databases out there. It has more than 30 cancers, more than 12,000 samples as of now, a lot of dimensions across different omics and clinical bases. And also you have databases like CCLE, which is the Cancer Cell Line Encyclopedia. You have multiple cell lines across different tissues and lineages. You have many, many different cancer subtypes within that, and you have drug responses and many other dimensions of data as well. So typically, patient databases and cell line databases differ in a very specific way. For patient databases, it is enriched in multi-omic datasets. For each cancer, we are talking about hundreds of samples, but they lack comprehensive and systematic drug response. And that's expected because you cannot just go on and say, oh, I have a pool of 100 or 200 or 400 drugs, and I'm going to apply them on patients and see how they react. There's a specific treatment scheme, there's ethics and morality and all of that. So obviously you can get drug exposure data for some of these patients, but not systematic drug response. If you're looking at model system databases, you obviously have both molecular profiles as well as drug sensitivity profiles. However, the catch is that you typically have smaller sample sizes. For example, CCLE across, I think, 15, 20 tissues, it has slightly more than 1,000 samples in total. And then if you need a full omics profile, you can get that only for 640 samples or something like that. So for each specific tissue or specific lineage of cancer, you were looking at in multiple of tens of sample sizes. So our overall goal is we want to learn the translation and relevance of certain gene signatures using rich multi-omic data. So you want to use both the patient and the model system data combined fashion. And then we use that learning to improve gene selection in outcome models. And the outcome can be very different. It can be patient survival outcome, it can be cell line drug response or other model system drug response or so on. So the bottom line is there's lack of sufficient samples and therefore sufficient statistical power in some kind of an outcome model. And we want to learn knowledge from other data and integrate that into this so that we can improve statistical power for selection. So how do we go on doing that? So we first need to learn relevance of genes. And for that, we look at the concept of drivers in the sense whether there is a significant relationship between whatever upstream genomic or epigenomic events occur and their resulting gene expression. And if you just Google driver gene or functional drivers, there's a class of different definitions that differ from one another in maybe small details. But we are broadly following the idea of this paper that talked in length about the biological idea of drivers. And then how do we go on to bring that into a statistical model? So our idea would be a multi-layer model where mechanistic models will be learning gene importance or relevance. And then there we use both patients and model system. And then we'll go on to build what we call outcome models, which is the second layer of our modeling scheme that will associate the genes with drug response. So that will say that, okay, these are the genes that are the associated genes with these drugs. And then that we can only build for model systems because we only have extensive drug response on them. However, in this model, we want to bring in the evidence that we learn in the mechanistic level to improve power. So here is our overall pipeline. So if you forget about this right part of this for a second, what we're doing is that separately from the model systems, we're using the upstream genomics and epigenomics information to synthesize evidence on the transcriptomics. And similarly, we are doing the exact same evidence learning on the patient datasets as well. These two sources of evidence are learned by these green lines, which we call mechanistic models. We combine these two sources of evidences and bring them in to calibrate what we call the outcome models here. And so these models are simply feature selection or variable selection type models. So you have one drug response maybe that you're focusing on. You have a bunch of gene expressions and you see which ones are associated. There, we calibrate these models based on whatever we learn using the mechanistic models. So obviously, because patient and cell line databases differ in terms of sample sizes and other features, this learning from the model systems using the mechanistic models and the patients using the mechanistic models as well differ slightly in terms of how statistically we formulate them. So that's what I'm going to go into next. So we need two versions of the mechanistic model, as I say. Patient cancers have obviously higher sample size, as I mentioned previously. So single lineage models are sufficient. You don't need to borrow strength. For typical TCGA cancers, you would have at least 1,500 samples. For most of them, you have 400, 500. And then there are cancers like the breast cancer, BRCA, for which you have more than 1,000. For model system databases, you have lower sample sizes. There will be some cases where one lineage may have only 20 or 30 samples. So you need to borrow strengths across related cancer. And you need to deconvolve different kind of effects. There might be some baseline differences between different lineages of cancer. There will be interaction of such differences with the true upstream effects, and then there will be the main upstream effects, whatever gene relevance you were learning from the previous step. So how do you go on building such a model? So this is how our multi-lineage model for the cell line looks like. So you have the gene expression, which is G. You're modeling that on lineage because you have samples from different lineages here. So you have some lineage main effect, and this F is very general. I will go on to formulate that more rigorously later. You have some upstream main effects, so U's are your upstream. So for each gene, you have some matched upstream elements, some matched copy numbers and methylation sites. And then you obviously have the interaction between these two. And obviously, then there will be some kind of errors. So how do you go about working with such a model? If you assume for a second that you know what F is and you know how to mathematically manage this. So first of all, you can build this model for gene by gene basis. So this is highly parallelizable and computationally advantageous. So first thing that we'll do is we will see if there is any baseline lineage effect. If that is not true, then you can ignore the interaction. And you can just go on to build a main effect model and test the upstream main effects and quantify that significance using some kind of quantifier like p-value or something equivalent in Bayesian models. If not, you can test both. You can test both the main effects and the interaction effects, and again quantify the evidence. So the choice of F is then crucial. And the simplest choice is to build a linear model. However, that is not necessarily a good choice because there's ample evidence in the literature and even in CCA data or CCAD data. If you just go in and you just plot some upstream methylations against some corresponding gene expressions, you will clearly see that they do not really behave linearly. Sometimes they're piecewise linear, sometimes it's not even that, it's more general. So you need a flexible specification of F that can accommodate highly nonlinear associations. And to make sure that, we chose Fs to be Gaussian processes. There have been previously works on splines and other such things, but there you have to specify knots and so on. So with Gaussian processes, you essentially specify each F to be a Gaussian process, and then you add them to make an additive Gaussian process, and you can do Bayesian inference on that. I'm not going into the details of that, but I'll be happy to answer questions on that at the end. And then single lineage models are simple. So these are the ones that we would build for patients, and here you only have the main effects. So again, one model for each gene, you can directly test for lineage main effects. The quantification that we use, just like if you used a classical linear regression, you would use a p-value. We use what we call pseudo-Bayes factors. So we use a Hamiltonian Monte Carlo-based procedure, and based on that, we compute these pseudo-Bayes factors, which are not exactly Bayes factor, but some approximation of that perform well. Again, I'm not going into the details of that, but I can explain that later if necessary. So overall, our scheme looks like this. So your multi-lineage evidence learning first tests for the lineage, then goes into the model with interactions if there is evidence of lineage effect. If not, it goes into the model with only the main effect. And then your single lineage evidence learning is directly testing for the main effects. So then we have learned the evidences from these models. So how do we build the outcome model? So statistically, this is a problem where you have some prior information on each covariate. In our case, these are the zero-based factors. You have a directionality of evidence, so you know that if they increase, you have stronger evidence for that gene. However, the other way around is not true. If you don't have any evidence for any gene from the previous layer, it does not mean those genes are useless. They might have association either way. Your data might not reflect that in the mechanistic layer. So the absence of such evidence does not mean futility. So you need some kind of information system for the model to learn of such data. And most variable selection or shrinkage approaches haven't exploded until very recently. There have been some discrete type procedures like group Leso, but not continuous. So in the last 2-3 years, people have tried to build Bayesian priors that can do this. So this is where the main novelty of our work comes in. We built this variable selection model where we are modeling some outcome. This will be drug response for us on the gene expressions, and we have these coefficients. The idea is that you build some prior on these where the evidences go in. So this is the prior that we propose, and we call it the calibrated spiking slab. So the spiking slab idea has been there for a long, long time. The idea is that you have essentially these deltas, which are the selection indicators. So if they are 1, then that variable is included. If they're 0, then it's not. And then the probability of that being 1, which is the probability of inclusion, has a prior. And that we formulate as this beta, where these E's are your evidence quantities, in particular, our PDFs. So the question now is, basically, how do you specify this F that takes the evidences to this probability scale? So the advantage of this are multifold. You are flexible in terms of how many evidences, what evidences you use. You can control importance of each dimension, and so on. The particular thing about this model is that with increasing sample size and also dimension, you need care in fitting such Bayesian models. So again, I am skipping some details there, but there are certain diagnostics that you can do to make sure your model is actually making sense. The particular evidence calibration map that we use to take the evidences from the PBF scale to the beta prior mean scale is this. So the idea is that there are standard cutoff ranges for base factors. There is particularly one Kaas and Raftari paper in 1995 that talks about these. So 0 to half or anything left to 0 is essentially not significant at all. Half to 1 is suggestive, then 1 to 2 is slightly stronger, and then decisive above 2. So our idea is that the prior mean of the posterior probability of inclusion starts at uniform because absence of information does not mean futility. And then we slowly grow at first, and then eventually, as you have stronger and stronger evidence, you put high posterior probability, high prior probability of inclusion on that specific covariate. And in the bottom, you can see the corresponding prior densities. So with that out of the way, we perform the integrative analysis of data. So we choose 3 TCGA cancers, the gynecological cancers, breast, one of the uterine cancers, and ovary. We took the corresponding cell line lineages from CCLE, and we took the drug response from GDSU. So after some cleaning and filtering with minimum sample size and variability and so on, we have these many genes and drugs. So for the cell lines, we are building multi-lineage mechanistic models. So there's one model for one gene. And then for the patient, we are building single lineage models. So there are 3 models for each gene, for each of the 3 TCGA cancers. And then we have those many drug response models. So what do we get from them? I'll briefly talk about the mechanistic model results first. So this is a plot that shows you how many genes were decisive across the different sample sources. And as you can see that the cell line alone cannot pick up a lot of signals that are being picked up by the other sources. So bringing in patient information is actually useful. We also tried to see how much fidelity these things have. So we went on to do GSEA using the 4 PDFs with higher activity within the gene set than outside. So this is the result for some of the GSEA analysis. And this is one particular gene set. Could you repeat that? Okay, so we have this particular gene set where you can clearly see that there will be some for which there's no source of evidence, or some for which the cell line model will miss the signal, but the others will let you know about that information. So that tells you that there's some information that you can know from this patient system. But how do you know that that's useful? That's where the outcome models come in. So we first summarize the outcome model based on variable selection, and we use FDR control at 10% level by treating 1 minus posterior probability of inclusion of a p-value type quantity. So this is a broad summary of all the drug response models that we did. And then we particularly try to look at if there are affinities of certain genes to certain cell line systems. So for example, here you can see that there are certain genes that are appearing more in breast models, more in ovary, or more in uterus. And then you can obviously go on to look at specific examples of these drug response models. We here show 2 models for the drug docetaxel in breast and cisplatin in ovary, and both are known drugs that have been used to treat those diseases before. And the crucial point here is that these red lines are selections, number of discoveries from our model at increasing levels of type 1 error. And the blue lines are the same for an uncalibrated counterpart of our Bayesian model. The point is that at the same level of type 1 error, our model allows you to make more discoveries. So you get more candidates that you can look at in a therapeutic sense in further investigations. So while being conservative and while controlling type 1 error, you are still getting more statistical power to identify signals. And that is the bottom line of this work. I have more of these results, but I obviously don't have time to talk about them. So I have a Shiny app set up where you can go and you can interactively play around with these results. To wrap up, our calibration method has benefits. It allows you more statistical power at the same sample sizes by integrating data from outside sources. There can be multiple future steps here. You can give this a precision medicine direction where you can say that, okay, my coefficients in the outcome model, the betas that you see, can be functions of even further heterogeneity or other kind of features. So there have been works on that before. This Hessian Tibshani work has been adopted to build varying coefficient type models with tumor heterogeneity data and things like that. There could be a joint modeling scheme to jointly build the layers instead of taking the outcomes from one layer and then putting them in the other. The one thing that I'm doing for my thesis right now is a joint variable selection. So what if we are doing separate drug response models, you could bring them together into a single multivariate as well as multi-layer model. And then you can also think about automatic estimation of evidence importance without putting the evidence importance in sort of that ad hoc way. So that's where I will stop. and I will take any questions that you might have.
Thank you so much. So now we start our question and answer section. So Kathleen, Carol, and Tess, do you have any question? Oh yeah, hi Carol.
Sure. I'll go. Rupen, that's really great work. So interesting, so complex. I'm an application person rather than a person who is applying these kinds of models. So I have a couple of questions. One is, you talked about how your model's better in terms of minimizing false discovery rate and maximizing discovery. How do you validate that? How do we know that's the truth?
Exactly. Yeah, that's a very good question. So I think the easiest way of doing that would be to start with some kind of a simulation study. So this work is more focused in a bioinformatics type way, and we wrote this paper for more biocomputing type journal. So we did not include the simulations here. I have another paper based on this work, which is called 5app, for which we are going to bring out a preprint very soon, probably in the next week, where we did extensive simulations on this. So what we tried to do is we tried to generate, say, first we tried to generate upstream data from some distribution that we already know. Then conditional on that, you can start generating expressions, gene or protein expressions, and so on. And then you can, conditional on that even, you can generate a further third layer, which would be some form of outcome. It could be continuous survival, something else. What you would then want to know is that if I tweak how those gene expressions are related to the outcome, if I have different combinations, say, for example, you have one gene expression which is directly related to the outcome, but there is no evidence before, versus there's one gene expression that has a lot of evidence, but it is not truly related to the outcome. How do you look at those things? So in those simulations, we exactly create scenarios like that, and then we evaluate our model versus the uncalibrated counterparts in terms of selection matrix. So you could compute AUCs, you could compute true positive rate, false discovery rate, and so on, and see how those performances differ. And there we see two things. First, not just our methods, but even a few other calibrated methods in general always do better than uncalibrated methods. They have always have better selection performance and so on. And further, the particular advantage that you have even in those scenarios is that if you are at a very poor sample size to covariate ratio, even then you can preserve at least some good AUC or some good selection metrics. So it's still not absolutely worth nothing. So that's the idea. That's one way by simulation that you could do. The other way to validate would be to do something like essentially something like a literature search. Like, say, for example, I'm building a lot of drug models, right? So one thing you could do is you could take all the drug hits, like, okay, these are the drugs and these are the associated genes I have. There are tools nowadays, there are packages and other things that allow you to search, say, PubMed or other existing literature databases to see if those hits are already known, how many literatures have mentioned those hits before, how many, and so on. And then you can use some empirical p-value computation based on that. That's something we have also thought of doing for our next work. So I think those two would be ways of validating such things.
Right. Thank you. That's that sounds reasonable to me. Congratulations on the great work. I'll pass it over to the other judges.
Oh, Kevin?
Rupen, that was a fabulous talk, I think, in building the model and telling us about it and the uniqueness and then the need for it. This might be very similar to Carol's question. I was wondering if you've actually run this for a ground truth or a known. So not in simulations and not necessarily in model building, but is there a ground truth that does not use your model that you can apply it? And have you done that to see how well it performs against the closest competitor model?
Yeah, so I think I'm not exactly sure what you mean by ground truth in this case, but like one thing that we did in our simulations at least was that generate the data from a model that is not exactly the model that we are fitting. So generate it from some other source model where the associations still are preserved, but they're not exactly generated from that Gaussian outcome or something like that. Okay. I think that's something we tried to do. And there we also tried to see whether the model can be successfully picking up signals. And we also did, just like, I think I mentioned this briefly in the answer to my previous question, but what you can do is you can try to see if the model picks up the true associations even when there's misleading data or something like that. Say, for example, you could generate a scenario where there could be genes that are not truly associated with the outcome at all, but you put in a lot of misleading evidence. That might happen in many cases. Like there might be molecular or cellular events happening at the upstream level that do not really express themselves at the outcome level. And you could generate those kind of scenarios, and you could still see whether the model for those specific covariates is performing well. So I think that would be one way to do that. I mean, it's hard to do that even for the patient data, because the best drug data you can get for TCGA patients is what is called drug exposure data, which I mentioned briefly in my talk. And the idea there is that those patients used to be somewhere in some hospital and they were given some drugs for their treatment. And you just know whether they reacted in a certain way to those drugs. There's no way to know whether that was an optimal decision, no way to know whether that was the best outcome for them, and so on. So it's hard to validate such things.
Now, I'm presuming everything here was human. Can we very quickly adapt this to mouse? Is it completely applicable?
From a theoretical standpoint, yes, because the model does not really care about which sources of data you have. It only cares about the fact that you have multiple systems. Now, by multiple systems, it does not really care about, is it patient, is it cell line, is it some mouse model, is it xenografts or other things, so on and so forth. So you can theoretically apply this model to any of those things. I think the particular care that must be exercised is, this is not a causal model. This is an associative model. So whatever signals you are looking at here are associations. All we can say, and I said this in the end of my talk as well, they can say that, okay, these are potential therapeutic candidates. You can look at this with further investigation, further cellular research and everything. But we cannot claim that, okay, this association means that there is a reason why this gene is coming up in this model. So that, I think, the interpretation of it is what you need to be careful about if you are applying it to other systems. Theoretically, there's no blockage in terms of applying it, but you have to be careful about how you understand those results.
Okay. I have one more, but I can let somebody else go.
Yeah, we have 1 minute left.
Okay.
Yes.
Sure. I have a couple questions, and they're more technical about the model. But sorry if I missed this. How did you determine which genes you actually used in the model? I know on like slide 16, you had like a list of like 5,000 or something.
So we were really lenient about that. So essentially, if you look at both TCGA and CCLE, I think the whole gene pool is like 20,000 something. And with the sample sizes of like hundreds, no variable selection model in the world is going to give you a good performance if you bring in like 20,000 covariates. So that's basically a nonsense thing to do. So what we started doing is just doing basic filtering. Like if you're looking at a specific cancer and you have a gene that does not even have enough variability across the samples you have, then you already know that that gene is non-informative. Whatever model you are going to do, that is not going to be picked up anyway. So you can remove those things. You can compute the variance or the coefficient of variation, and you can put a cutoff on that, saying that anything above this, I'm only. You can check data quality, like missingness and other things, and similarly put a cutoff on that, and so on. So that's the kind of filtering that I did. And then eventually for each specific drug response model, you can do something further. You can build univariate models for each gene. So one drug response, one gene expression. If it does not come up significant in that univariate model, there's no way it can come up significant in the multivariate linear model. Like you can theoretically show that. So you can do some further filtering there as well. So we did those basic filtering steps. We were very lenient again in terms of our cutoffs on all. That's why we had quite a few genes, because we also wanted to see that our model can handle high dimensionality. But I think that's typically how we do it.
Well, I believe this concluded our question and answer session. Thank you so much for the presentation. So let's move to our next presenter, Dr. Kathy Chan. Please go ahead and share your screen, and thanks ahead for your presentation.
Okay.
Right, perfect.
Oh, okay. Thanks, Dr. Leung, for the kind introduction, and thanks to everyone at EMGS for organizing such a great event. My name is Xi Chen, and I'm a staff fellow from Division of Bioinformatics and Biostatistics at the National Center for Toxicological Research at FDA. I will be presenting on Animal Gain, a generative AI alternative to animal clinical pathology testing. Before I really start my talk, I'd like to give you a disclaimer. The information in this presentation represents the opinion of myself and doesn't necessarily represent NCTR's or FDA's position or policy. Okay, clinical pathology testing of laboratory animals in preclinical studies is crucial for drug safety evaluation and risk assessment. However, the conventional testing process is expensive, time-consuming, labor-intensive, and limited by small blood sample volume. Especially for the small-sized animals, we can even not get enough blood sample to test all the parameters we want. As the toxicology community and regulatory agencies are moving towards replacement, reduction, and refinement of animal studies, and meanwhile, abundant animal data are available from the public domain and private practice, we are exploring an artificial intelligence approach to learn from the existing animal testing results to generate the new animal data without conducting additional animal experiments. This so-called animal GAN is a generative adversarial network-based framework. The GAN model is a generative model that can produce new data with similar distribution to that of real data. It is composed of two models, namely generator and discriminator, which compete each other. Our animal GAN model will receive treatment information, including molecular descriptor, time point, and dose level of compound as model condition. At each training step, as the generator tries to produce clinical pathology data that fulfill the specific treatment condition and more similar to the real data, the discriminator becomes better in distinguishing between the generated data produced by the generator and the real data from the real compound treated animals. When this GAN model was optimized, we can use the generator to produce animal data. The data set we used in this study is extracted from the repeated dose studies in the OpenTGS database. We have more than 80,000 compound treated rats, which treated by 138 compounds at 3 dose levels and 4 different time points, corresponding to 16 and 49 treatment conditions. And for each rat, 38 clinical pathology measurements were tested, including 17 hematological measurements, such as the red blood cell counts and hemoglobin, and 21 clinical chemistry measurements, such as ALT, ST, ALP, and total bilirubin. These measurements are very important to assess liver injury. To use the chemical structures of compounds as model input, the numerical representations of chemical structures were calculated using a Python package named Moderate. And the Moderate can calculate 1,826 molecular descriptors for each compound, including both 2D and 3D descriptors. With these data, we can begin to develop a model. We split the data set into two sides, using about 8%, 110 compounds treated rats under 13-17 treatment conditions as training site to construct the GAN model. This is an iterative process, and at the end of each training epoch, we use these criteria, including the convergence of the loss curves, cosine similarity, root mean square error, and a Disney plot to evaluate if the generated data and the real data follow similar distribution. And this figure demonstrates the evolution of model performance during training. Clinical pathology measurements of rats, even exposure to different chemical treatment conditions would have a moderate similarity. To justify the generated data could represent the real data. Cosine similarities and root mean square errors between every two treatment conditions were calculated respectively to estimate their background control distribution. At 6,000 training epoch, all the criteria were passed and reached their best. So eventually, the model at 6,000 training epoch, at 6,000 checkpoints, was selected as the animal GAN model in this study. Then we asked the animal GAN model to estimate the clinical pathology testing results for the 332 treatment conditions of the remaining 28 compounds in the test site. Due to those are unseen data in model construction process, so we can use them to evaluate the performance and the generalization of our model. And here are the results. Cosine similarities between the generated data and their corresponding real ones are significantly higher than the background controls. And the root mean square errors between the generated data and their corresponding real ones are significantly smaller than background control. And the t-SNE plot also demonstrated there is only subtle difference between the distribution of the generated data and the distribution of the real laboratory testing data. So these all demonstrate that from a statistical perspective, we find a good model. The final goal of this study is to generate high quality and efficacious clinical pathology data, which can achieve equivalent performance in toxicity assessment, just as the real animal testing results can do in toxicity assessment. So, and the clinical pathology data, we usually use them in toxicity assessment through identifying the treatment-related changes in clinical pathology measurements. So we investigated whether the animal GAN generated clinical pathology data could be used to identify treatment-related changes. Specifically, for a given treatment condition, we compared the difference between the vehicle control treated group and the compound treated group for each measurement to assess if there is change between the two groups. Those comparisons were done by the generated data and the real data respectively, and then we compare the assessment results to see if we can obtain comparable results. If the test results based on the statistical testing based on the generated data can the test results be based on the generated data and their corresponding real data were both indicating there is no change or there is change. Both this demonstrate there is no change between the treated group and the control group, or both indicate that there is change in some parameters. We will consider that the generated data and the real data have the same power in toxicity assessment. Otherwise, we consider they are not consistent. So this is the comparison results. The green cells represent the consistent evaluation results. Most cells in this figure, we can see they are green, so indicating the high concordance between our animal GAN generated data and the real data in toxicity assessment. Zoom in the results to see each measurement. For every measurement in different category, we all get the high concordance results, and specifically, the concordance on hepatotoxicity related measurements are all more than 96%. We think this is a very good results we want. And the concordance on nephrotoxicity related measurements are even higher, indicating our animal GAN model generated clinical pathology data can achieve equivalent performance in toxicity assessment as the real laboratory animal data candid. To further validate the proposed animal GAN model, we employed another large publicly available database, drug matrix. We extracted the clinical pathology testing results from the compound treated and vehicle treated male rats from the drug matrix database to do the evaluation. We asked the animal GAN to generate clinical pathology data for treatment reported in drug matrix and use the generated data to assess toxicity as we did before in the TGCase test site. Due to our model were developed using TGCase data, the rules we learned are all both based on TGCase. So we'll use the vehicle control treated group in TGCase to identify treatment-related changes for the generated data. Due to the variations induced by different, the experimental protocols and the environments between the drug matrix dataset and the open TGCase dataset, we constructed a benchmark first for fair comparison. We extracted the clinical pathology data from the two datasets for the overlapped treatment conditions and compared the toxicity assessment results of the overlapped treatment condition from drug matrix and open TGCase. We found that for the treatments reported in drug matrix, the generated data can be used to assess toxicity similar to how the experimental data is used, with the average concordance rate around 70%, which is comparable. The animal GAN generated data and is comparable with the concordance of toxicity assessment results by using TGCase data and the drug matrix data for their overlapped treatments. So in summary, we propose a GAN-based framework named Animal GAN, which learns from the existing animal data and holds promise for generating new animal data just from the chemical information in combination of the dosage and the treatment durations. Learning from the pre-existing data from TGCase, Animal GAN successfully estimated the clinical pathology testing results with high similarity to the corresponding animal testing results. Animal GAN estimated clinical pathology testing results can be used to assess toxicity, and the assessment results are highly consistent with the animal testing data-based assessment. Finally, I would like to take this opportunity to express my gratitude to my supervisor, also my division director, Dr. Vida Tong, and my postdoc supervisor, Dr. Zhuchao Liu, who just take his new position in iTalks, for their tremendous support and great help during this project. And many thanks to our collaborators, Dr. Scott Albach from BNTP at EHS and the Dr. Ruth Roberts, who is a co-founder of Apconics and a professor at University of Birmingham, for their time and contribution to this work. And thank you for your attention. I'm happy to take any questions.
All right. Thank you so much. All right, so now we have our question and answer session. So any of the judges have questions?
Sure, I can start. This is really incredible work. I love Animal GAN. I've heard about it before. I've got some echo here. I'm curious about a couple of things. First, so you basically, when you're building your model, you put in all the clinical data that you have from the animals, as well as the gene expression data. And I'm curious as to whether you've looked to see what the gene expression data actually added. Did it, if you ran it just on the clinical parameters that you measured in the animals without the gene expression data, how much is the model that much worse? Or is the gene expression data really, really increasing the power of the model?
Oh, in this animal GAN model I presented today, we just use the clinical pathology data, but I think you may heard the Animal GAN before. This is not the first GAN model we built. Before this study, I built another GAN model. We called it ToxGAN, but the similar concept of this study. In that study, we used the gene expression data.
Oh, okay. So this study, you didn't include gene expression
in
data
this study, we only use the the clinical pathology testing measurements, such as ALT, ST that way.
Okay, that I sorry I missed that part of it, because and then it kind of answers my next question then, which would be, what can you use in vitro models? Can you build this into the system at all? So I guess it's not relevant since you're using clinical outcomes in animals. Okay, and I think this is it's very cool. So you can basically have a very good prediction of a potential outcome in an animal based on your model without actually doing any of the testing.
Yeah. Our rationale is, generate data in a less expensive way. So yeah, we can save a lot of animals, actually the compound.
Yeah, great. So is this being used for drug screening or anything right now? Have people tried it? Are people applying it yet?
We did the exploration ourselves, but I didn't know if there is someone usually used in the drug screening. No, I'm not sure.
Okay, I was curious. Are you going to make a tool available? So there would be like a tool where you can enter.
We already make the ToxGAN model. I mean the gene expression based GAN model available, and this is a non-published work now. We will publish it.
Right.
I think in a few months.
And so you'd produce the R codes, put it on GitHub or something. I'm just wondering if you're thinking about a software tool that non bioinformaticians could use.
Yeah, we have a plan to do it, but for now we didn't have the software now.
Right.
But we already make the source code in Python available in the GitHub.
Okay. Thank you. Great work.
Thank you.
Okay, I could try a question. I was really interested in the learning process, the epochs and the training that was going on. How did you define an epoch?
Define the epoch. Okay. When all the training data were used in the model, we call it one epoch or epoch.
So when you used either the word tests or training, you went up to 6,000.
Yeah, training 6,000 epoch. Actually, we training million thousand, and to find at which epoch we can get an optimized model. So in my slides, let me find that.
I was trying to figure out the relationship between the training and the epoch. And then I was curious to hear what you thought happened when you went to 7,000 tests. Yeah.
Yeah, this figure demonstrates the evolution of the model performance during training, not about the test, just the training. When we training, we will use the training set to run a lot of epochs and use some criteria, including the loss curve and the cosine similarity RMC and the testing plot to find a good checkpoint. That's the training process.
Yes. So what happens between 6,000 and 7,000?
6,000. For example, for the cosine similarity, we can see at the 6,000, the cosine similarity, we can say it reached a higher position than other epoch. So we choose this one. The cosine similarity at 6,000 training epoch will higher than the 7,000 training epoch, and the RMSE will smaller than other epoch. So we choose this checkpoint.
Now, between any of the epochs, did you do anything that is sort of a disruptor, maybe to prevent any overfitting or
overfitting? Okay. Actually, the GAN model the concept is the compete between the generator and the discriminator. So the loss curve will always up and down, not like the predictive model. So yeah, when we also add it's a bit the statistical. We add a regulatory term in the loss curve, so to avoid overfit. And also when we use the test site, we will test to test. This is the unseen data. If we will have a good performance in test site, so we can say the model have the generalization ability, not overfit.
Okay.
Uh-huh.
Okay, thank you.
Thank you.
Does anyone else want to jump in with the question, or I can ask one?
We have 30 seconds left, so really quick one.
Oh, okay. I had similar questions. I was wondering about overfitting because I saw that your p-value is like minus 200 something when you're comparing like the simulated data versus the actual data, but I guess you just kind of answered that question.
This one?
Yeah. But I'm not a modeler, so maybe that isn't what that's showing, but it just seemed like it was like really generated versus the real data. Yeah, I guess that's not a clear question. We can move on if you want.
Okay. I guess that's concluded our question and answer session. So thank you so much, Dr. Chen, for the presentation.
Thank you all.
Thank you so much. All right, so we will move to our final presenter. So Dr. Lee, would you mind sharing your screen?
Yeah, thank you, Dr. Leung. Yes. Can you see my slides now?
Yes, it's perfect. Thank you.
Oh, thank you so much. To secure, I can give this talk very fluent, so I turn my video off. So thank you. First of all, I really appreciate all of you to give me this opportunity to present my work here. This is Ting Li, and I'm also a colleague of Shi Chen, and we are working at the same division of Bioinformatics and Biostatistics in the National Center for Toxicology Research. So today my talk's title is Deep Learning Powered Drug-Induced Liver Injury Prediction using the model level representation. So before I really give my talk, I would like to have this disclaimer first. The information in this presentation only represents my opinion and does not necessarily represent the NCTR or FDA's position or policy. Here, I give you an overview about the drug development stages. Before the drug can go to the animal safety evaluation, that including 4 previous stage steps, including identify the target, identify the compound that's working on this target, and establish the activity profile and select the clinical candidate. And then once the candidate successfully passed the animal study, usually takes around 5 years, it can go to the clinical trial. From phase 1 to phase 3, more and more humans are exposed to this candidate drug to evaluate the safety and efficacy profile of this candidate drug. And once the candidate drug is successful from this clinical trial phase 3, it allows to go to the market. And there is a phase 4 called the post-marketing studies. In this part, it is going to monitor the safety profile of this large population. So to successfully launch a drug from the very beginning to the market usually takes around 12 years and costs $2.6 billion. However, 90% of this clinical trial failed, and then 30% of them were due to the toxicity. Here on the figure on the left side, summarize the toxicity causes of the failures in the preclinical trials. And then we can find that hepatic, which is called drug-induced liver injury, is one of the leading cause of these drug failures. One of the reasons is because DILI risk share the low concordance between the animal studies and our humans. That means alternative methods are important and needed to improve the translation of the preclinical findings to the clinical applications. Actually, there are decades of effort have been made to find alternative approaches to improve the toxicity prediction. And in the recent years, there are software claim that they can improve the toxicity test. Besides the research community effort, actually the regulatory from both Europe and the United States, they also promote alternative approaches to improve the toxicity prediction. For example, from the REACH program, they promote the 3R principles for the animal study, like the replace, reduce, and refine the animal study. So we want to utilize this artificial intelligence to transfer the in vivo study into an in silico study. Then in the recent years, deep learning had so many successful applications in the fields such as computer vision, speech recognition, natural language processing, and medical science. And for example, the artificial intelligence system can be used to screen the breast cancer, which can achieve like a comparable or even better performance compared with our human experts. So inspired by these successful stories, we also would like to utilize this deep learning algorithm in our DILI risk prediction. So when we're talking about deep learning, that means we need to a large data set. So we obtain the DILI labels from the DILIst, which is a large binary DILI classification data set for the human hepatotoxicity to mimic the real-world scenario that only the previous accumulated knowledge can be applied to the future ones. So in the first step, we collect the drug approval year information from these three resource, drug bank, Wikipedia, and FDA label. In total, we get 1,002 drugs had their approval year information available. And this drug approved before 1997 was used for developing the model, and the rest of this drug was used to test the model. So here is a framework for this deep DILI model. The basic idea here is we want to input the chemical descriptors into this deep DILI model and let this deep DILI model to make a classification for these compounds, whether it will cause liver injury on the liver or will not cause liver injury. So usually a classification model including one algorithm, but here we use 5 different of them because we had this hypothesis that different machine learning models could capture essential information related to different DILI mechanisms. And for each algorithm here, we developed 100 these classifiers, and the predicted probability were collected together at this model level representation. And this model level representation would further fit into a neural network. Actually, this format is very similar to an ensemble method, but here we use a neural network because we get this hypothesis that the neural network could harness the individual machine learning model to improve the prediction power. So we want the neural network to make the final prediction of the DILI risk of the compounds. And so in this study, we have investigated 3 different chemical descriptors, including MOR2, MACCS, and the MOR2 WEC. The MOR2, including 777 physical chemical descriptors, which was encoded from 2D chemical structure information. And the MACCS is a fingerprint-based chemical descriptors. It includes 166-bit binary information to indicate the presence of these predefined substructures. And the MOR2 WEC is a chemical representation generated from an unsupervised pre-trained machine learning model, which was trained from almost 20 million of compounds. And it can generate 300-bit long vector representation for the molecular substructures. So in this study, we aim to answer 3 questions. The first question is, could this delay potential of this newly approved drug be predicted by accumulated knowledge of the early approved drugs? What means early approved drugs in this study means the drug approved before 1997. The second question we ask is this model level representation could be more informative than the molecular-based representation for delay prediction? The third question we ask is, could this deep delay be better than the published delay prediction model? So for the first part, we developed a 3 delay prediction model from the descriptors MOR2, MOR2-WEC, and MACCS based on the drugs approved by before 1997. And these 3 models were further evaluated on this test set that the drug were approved by and after 1997. And these 3 models were evaluated on these 3 performance metrics, including Matthew correlation coefficient, accuracy, and AUC. And we can find that the model developed from the MOR2 outperformed the other 2 models, which means this also helped to show that the delay risk of the newly approved drugs can be predicted by the model developed from the drugs approved before 1997. Then for the second question, we developed the other 6 different delay prediction model with the molecular-based representation. Here we use the MOR2 descriptors, and then we come including the deep neural network, logistic regression, KN, and support vector machine, random forest, and XGBoost. Then we compare these 6 models with the deep delay models through this matrix evaluation performance. And we found that deep delay outperformed the other 6 models, which indicating that the model level representation is more informative than the molecular-based representation for this delay prediction. Further, we ask the questions whether this deep delay model compared with the published DM model. Here we colored this published DM model in yellow, and then we compare this model on 3 external validation data set, including the NCTR data set, Greening's data set, and Schu's data set. For each data set, we evaluated them on the 3 performance metrics, including accuracy, sensitivity, and specificity. For example, if you see the accuracy, we can find that the deep delay model developed from MOR2, which colored green here, had a better performance than the NCTR data set and the Greening's data set, but with a fair performance on the Schu's data set. Further, we did a delay application to screening the delay risk for the compound from the drug bank. In total, we have screened around 10,000 for compounds. But these compounds, we don't have the ground truth of the delay risk. So we plot the predicted probability values yielded by this MOR2 deep delay model to show the distribution of these prediction values. And we found that the majority of these compounds either go to the left side, indicating that the less likely delay risk, or either go to the right side with a high delay risk, and the comparable small number of compounds located in the middle that with uncertain of the delay concern. So the take-home message for this study, the first one is, compared with this molecular-based representation, the model level representation improved the performance in the delay prediction. The second part is this deep delay model could serve as a screening tool for delay risk of the compounds in the preclinical setting to improve the delay risk prediction. The third one is this framework of this deep delay framework actually could apply to other toxicity endpoints such as carcinogenicity and mutagenicity. I will take this a moment to give thanks to my supervisor, Dr. Wida Tong, my technical lead, Dr. Zhichao Liu, and my colleagues, Steve and Leopold, and my collaborators from Dr. Ruth Robert and Dr. Shraddha Thakur. And thank you so much for your attention. I'm happy to take your questions.
Well, thank you so much for the presentation.
Yeah,
okay, so thank you so much. So, okay, so we will have our question and answer section now.
Yes.
I guess I'll go first. I have a very general question that maybe we were all thinking is, I know this is used for drugs, but could it be used for like environmental pollutants to look at different endpoints?
Yes, we definitely can use this model to predict the daily risk for the environmental chemicals. We can do that, like to give information, like to priority the daily risk for this. Yeah, definitely sure we can do that.
I have a question, I guess. Kathleen, you go first. I'm always jumping in there. You go.
I was
Go on.
afraid if I really wanted to make sure Tess went first for once. So in the accuracy classifiers, so I think it's really, really cool that they're used in combination because I hadn't seen that before. We use them all separately. What did you do to choose the ones that you had and the number that you have?
What do you mean?
So you had 5 accuracy classifiers. Why 5 and why those?
Oh, so basically at the beginning, we want to include in very simple, like the KN, and we also want to including the linear regression related model can cover the linear part, the logistic regression. And we also want to see the decision forest, like the random forest, which is a robust model, and the XGBoost is the most popular model as well. So we want to cover to including the models from different perspectives, so we can cover the capture the delay mechanisms. Yeah.
And then I saw that your particular model did better, but it's not that much better. Were you expecting more?
If we only look at the performance, yes, it is not that great improvement, but we need to see the performance correlated to the endpoint that we are studying. So delay, drug-induced liver injury is a very hard endpoint. So I'm not expect to improve a great improvement, but we see they still maybe have an improvement can be made. Yeah.
Right. And did you ever test your accuracy classifiers, let's say 3 at a time, 4, 5, just to see
the like improvement, increasing improvement? I think you gave us a very good point to like you want to see whether I just add 1 classifier and 2 classifier, how this classifier can contribute to the final model, right?
Yeah.
Yeah, but I think it's a good way to try. But for right now, we just did the 5 at the very beginning, to want to including the as more comprehensive algorithm as possible. Yeah.
Yeah. Yeah. Okay.
Thank you.
Thank you.
Mm-hmm.
Yeah, congratulations on this fantastic work. It's absolutely beautiful. I loved your introduction too. Those introductory slides were really cool.
Thank you.
I'm curious. So DILI arises through a variety of mechanisms.
Mm-hmm.
Is there one mechanism or a subset of mechanisms that are working better in this model? Are there some mechanisms that you're just not predicting? Did you drill down into the data in that way, or?
So the mechanism, right? The input here is the chemical descriptors. And so when we're talking about the mechanism, then we maybe prefer to talking about the biology mechanism. And I think you give us a good perspective to thinking the model. How what we did is to look at the ATC class of the drug, which ATC class we can predict better. But we did that part, but I didn't show it here.
Okay.
Yeah,
because I was curious because there was a group that you predicted correctly, and I wondered if there were any similarities shared amongst the drugs that you correctly predicted versus those that you didn't predict correctly. Whether could you mechanistically tease out what works well in your model and what doesn't?
Yeah, we do that part, but I didn't show it here.
Oh, okay.
Yeah, thank you. Yeah, this is a great point to present next time.
Yeah, I mean, anyway, perfect. Thank you. So again, this is a really important tool for potential drug screening before, you know, to help prioritize what goes into the development pipeline. So congratulations on the excellent work. I'm imagining that you could pair this with early in vitro studies and have an even better predictor. Would that be something that you might think about in the future?
Yeah, I think right now what we are planning to make this as a tool that can easy for users to use. Like you can give me just a SDF file, and then I will make a prediction on it. And if once we can collect more data, and from which maybe we can make some more improvement from that part. Yeah, definitely good way to go in the future. Thank you.
Yeah, thank you. Congratulations to the team.
Thank you. Yeah.
Okay, we have 2.5 minutes left for the Q&A. Is there any further question from the judging panel or from the audience? If you guys have any question, please do fill in the chat box as well. So we have 2.5 minutes left.
Did my talk is so simple, so you don't have any questions?
Yeah. Okay. Yeah. So if there's no further question, we would like to thank all three presenter and their team for the excellent presentation today. Thanks a lot for your time to share your work with us. I really appreciate your participation. And also thank you all judges and all everyone in the audience for tuning in to today's event. Thank you so much and have a wonderful day.